Good morning, one and all. This is Debasish, currently pursuing PhD at Chhattisgarh Swami Vivekanand Technical University, Chhattisgarh, India. My presentation topic is Advanced Research and Conceptualized Insight to Herbal-Derived Medicine System, a modern approach for safer and reliable drug system. So nowadays, there is a renowned interest in drug of natural origin simply because they are considered as green medicine that are supposed to be safe. And the factor that emphasize this attention is the incidence of harmful nature of synthetic drug that is regarded as harmful to human being and environment. The advantages of natural drug over the conventional synthetic drug are the easy availability, the economicity, and lesser or no side effect on the health system as compared to the synthetic drug. In this current presentation, I am going to discuss about the various areas or the various prospects in the herbal drug system based upon pharmacogenomics, reverse pharmacological approaches, informatics or information technology integrated to this medicine system, AI and machine learning in Ayurveda for authentication of the drug material, anthropogenic and agroquality challenges that have been faced by this herbal drug system, and the biotechnological prospect to explore and enhance the herbal drug system. Coming to pharmacogenomics, so it is an integration of the principles of herbal medicine system with genomics, where the central concept is oriented in predictive, preventive, and personalized medicine. So personalized medicine is its concept that is very, very different from the concept of one drug fits all. So here the medicines is a personalized one that is being integrated with the genomic system of the human being or individual to the drug. So the ultimate goal is to attain the personalized medicine system or for right treatment to right person at right time, clubbing together the genomic structure, genomic integration of the human being along with the drug response. So as a part of the information technology assisted medication to patient to provide them safe, quality and patient centered care with the help of both doctor and patient, and they can keep record of ongoing process, medication and follow-up of the medication prescribed that will promote a good healthcare system and a good coordination of this type of integration of genomic profile or genomic template to the drug. Coming to the reverse pharmacological approach, in this current study of drug discovery and development, this is a very costly one, time consuming, less productivity, and yields low return on heavy investment. Despite there are many advancements in the technologies of genomics, proteomics, and metabolomics. So the new paradigm for drug discovery and development is the reverse pharmacological approach that will help the traditional clinic experience, observation, and available data on actual use on the patient as the starting point for the pharmacological prospect and study. Since the safety of the material used in the traditional system is already been established from the traditional use of track record, pharmaceutical development, safety validation, and pharmacodynamic study that have been carried out in the controlled clinical studies. These traditional knowledge and experiential database this will provide a functional lead to reduce time, money, and toxicity. So these are the main three orders in the drug development. And natural products offer a vast potential for novel phytochemical with a clinical activity. With the advancement of time, with the advancement of the research, multidisciplinary got integrated into the herbal system of drug and get coupled with transdisciplinary. And despite this possibility, they can be also integrated to different academics, to different clinics, and traditional practitioners. Those who can give the idea, those who can give the observations for the patient what they're treating for. So this will give infrastructure for this particular reverse pharmacological approach. Coming to the use of information technology. So with the advancement of the opportunity and globalization, the need for integration of information technology has created a challenge not only for the practitioner but also for the system as a field. So the available ancient text, the research articles, the experiences that has been engaged from different traditional practitioners as well as the current practitioners who are going for the use of this herbal drug system, their experience, their information should be integrated as, or should be clubbed into a particular data system where we can generate different types of data, where we can generate different types of information in regards to the disease as well as the drug system. The opportunities and challenges are also there, such as globalization, patent, intellectual property right, and biopiracy. These should be controlled by the modernized approach in the line of development of IT and innovation. Information technology is a ever-changing field, but there has been a trend that will provide opportunity for integration of latest technology with the principles and practice of this medicine. The major challenge is bringing cutting-edge technology that lies in the core of the practice, staying true to the principle, methodology, and practice, as well as with the patient care. Another challenge refers to the acceptance of new technology by this old system of medicine. As we all know, Unani or Ayurvedic, whatever you say in Pakistan or India or any type of system, these are very, very ancient techniques, or they have been described in very, very ancient system. So this information technology should integrate the should have a clinical study. They should have study on the drug development. They should have study on the analytical drug. They have manufacturing, quality control, and commercialization. So these Ayurvedic, right now, these Ayurvedic or Unani system that are being pursued in academic, clinical, and research field also get integrated into the information technology so that everyone will be benefited from this system. Coming to another modernized approach for identification and authentication of the drug material that are the raw material for the herbal medicine system. So DNA barcoding is an approach for the novel prospective tool for the taxonomist and unreliable alternative for morphological identification, which have greatly transformed the species identification process. Recently, AI-based machine learning approach has reported as a precise sequence analysis platform to authenticate the species identity. Now you can see the flowchart of machine learning based on plant phenotyping. So here you can use the image as an input, and here the image as input is being adjusted, and there is a segmentation, there is a region of interest of detection where we can detect the different um, prospect or the different raw material based upon their extract parts such as flower, leaf, fruit, and going for the phenotypic trait extraction based upon the size, shape, color, growth rate, diseases, etc. And we can get the result output. So here using artificial intelligence or machine learning for identification of herbal medicine system. So integrating this huge analysis of data into the machine or into the computer can lead for the easier authentication or identification of the drug material or the raw material. Coming to the various challenges that have been accepted or that have been accepted for the herbal drug system are based upon mainly anthropogenic and climatic challenges. So there are about in India, I am talking about India. India has about 44% of the flora which are used medicinally, and one of the 12 mega diversity country harboring two unique biodiversity hotspots, out of which 18 hotspots worldwide. Means there are 12 mega biodiversity are there, hotspots are there out of 18 hotspots that is totally present in the world. These hotspots are due to various agroclimatic conditions in Indian subcontinent. This will have a conclusive or conducive effect on the growth of variety of medicinal plant and other organisms. So nowadays, what happens due to the stress or due to this anthropogenic interference into this system due to forest fire, grazing, rainfall, heavy snowfall, wind, UV radiation, sun exposure, landslides, snowslides, soil erosion, tourism, and other anthropogenic inhabitants. So there is a loss or unavailability of these hotspots and this plant material. So here there is a means what we could tell as there is we don't find any authenticate or genuine drug material, and what happens in long run we need a good amount of raw material, but there is no raw material that is present in the system. So what the people do is to adulterate those raw material with other parts of the plant, thereby reducing the one quality and efficacy as well as the safety of the drug material. This led to high level of adulteration that definitely imposes a adverse effect on the efficacy of herbal medicine system. Now, coming to how we can counter this type of challenges. This is through a biotechnological prospect. So there are about 8,000 species of medicinal plants that are being used for medical purposes. With the demand increasing rapidly, many of them are being endangered or threatened, as I have told you. So also due to this more and more anthropogenic interferences into this ecosystem, they have been destroyed or they have been limited in the growth. So studies on the medicinal plant indicate that the bulk of plant material is obtained from root, whole plant, fruits, seeds, and bark, which are essential for the survival and regeneration of the same. Many of the important useful species are on the verge of extinction due to exploitation and habitat destruction. More than 95% of the medicinal plants are collected from the wild. There is a need to encourage multiplication and cultivation of this plant. So there are two approaches where we can do the conservation. One is in situ and one is ex situ. The in situ of the conventional method is the regeneration of plant in their own environment. This is time consuming that requires several years to grow and reach the point of desired time where or desired age where they can produce the desired metabolites. Ex situ conservation is the relocation of endangered or rare species from the natural habitat and keep it in the protected area that is equipped for the protection and preservation. Another prospect is that we can go for plant tissue culture. So plant tissue culture is the in vitro cultivation of plant cells or tissues under aseptic and controlled lab conditions in well-defined nutrient media to regenerate plant or parts of the plant for the production of the metabolite. Along with that, the use of bioreactors, or biofermenters by using the plant cell materials or the suspension cultures in the bioreactor or biofermenters can be used for the production of biometabolites. So this is all about my presentation. Hope you understand some of the point of the use of and analyze the herbal drug delivery system, or there is a need for the study of new drug system or plants where we can use those biometabolites into enhancing the safety efficacy of the new drug system. Thank you for your presence and persistence.